Good morning and welcome to the second session of Life Sciences for Winter School. It is 8 o'clock, so I will get started. Today we have our last 2 hours in the Winter School program. We will be continuing with responding to the environment in humans. Yesterday we discussed the nervous system and focused on the brain and the spinal cord and the reflex arc and all the neurons. And today we're going to move on to the eye and ear. And if we have any time available, we'll go back to the questions we didn't complete yesterday. I am Katie Lewis and I'll be your presenter for today. So please ask that you've got your learner material in front of you with all the questions, as we will be working through that today. All right, so before we go on, I'm just going to talk about the exam guidelines. So because today we are focusing on the eye and ear, this little table is taken straight from the exam guidelines, which you can find online. And it tells you everything you need to know for the eye and the ear before you walk into your prelims and before you walk into finals. So I always say treat it like a little checklist so that you tick it off once you've studied so that you know what it is. So for the eye, we're going to look at the structure and the functions of the eye using a diagram. So that's very important. Then you need to know what binocular vision is and its importance. You need to know the processes of accommodation and pupillary mechanism. And then they like to tend to focus now on visual defects. And there's 4 that you will need to know. And then if we move on to the human ear, again, you need to know the structure and function of the different parts using a diagram. Then you need to know how the ear is involved in hearing and balance, and then also hearing defects. Again, like I said, they are asking a few questions with the defects in the eye and the ear lately. So remember, the whole section of human responses in the environment makes up 1/3 of paper 1. And there are a lot of processes. So it's important that you spend time going through them and that you fully learn them and understand them. That is key before you enter your exams. Okay, so let's start off by going through the eye. So I'm just going to do a quick recap for you before we start the questions, because it's important that it's fresh in your mind before we answer. All right, so if we look at the basic structure of quite a few labels that is part of this, and like I said, it is important that you know what the functions are. A lot of the questions for the eye and the ear are also know the knowledge and then apply it to questions. So essentially, what will happen if this is damaged or if this is there? But we'll get to that later on. All right, so if we look at the structure of the eye, I'm going to start off with the cornea. So the cornea is this front section of your eye, and you can see it's convex. And convex means it bulges outwards. And why that is important is because it will refract light rays. And refract means it bends light rays. Remember, light rays travel in straight lines. And when it goes through an object of a different medium, it will refract it. But the more curvature there is of a structure, the more refraction will take place. So the cornea is convex, we say, and that will allow it to refract light rays. And it is also transparent, which is important because we want the light rays to basically go through the cornea and right to the back of the eye, to the retina, where your photoreceptors are. Okay, so that's the cornea. Then we're going to move to the back layer, so the outermost layer at the back, which is the sclera. So the sclera is just a tough, inelastic layer, keeping the shape of the eye. And also, if you look at a person's eye, the white part is the sclera. Then we move on to the choroid. So that's the dark blue layer. Do not get confused by chorion, which is in the reproductive system part, versus choroid with a D. So this is the dark pigmented layer. and it has blood vessels that is supplying the eye with nutrients and oxygen. But also what it does is it absorbs any excess light rays, because you don't want the light rays to bounce back, because that will distort the image that is being formed. So it's absorbing the excess light rays. So that will be a choroid, the dark blue layer. All right, then I go on. So if we look at a person's eye, the dark circle in the middle, that is called the pupil, and that is just a hole. And like I always say, you can actually, if you had to stick an object, a pencil into the eye, into the pupil, it'll go right through, because it's just a dark hole. There's nothing, it's not actually a structure there. You actually see in the back of your eye. And then surrounding the pupil is the iris. And the iris is, if you look at your eye or someone else's eye, is the colored part of your eye. Now, the iris is very important because the iris is controlling the size of the pupil. So it's making the pupil either bigger, which means it will dilate, or it will constrict the size of the pupil, making it smaller. So your iris, with its muscles in it, controls the size of the pupil. They often like to ask that question, what controls how much light enters the eye? And yes, light is entering through the pupil, but what controls how much light enters it would be the muscles in the iris. And we'll go on to how that works just now. Okay, then we look at the ciliary body. So the ciliary body has muscles in it, which would control the curvature of the lens. So will control how convex it is. Then you've got your suspensory ligaments. So that's the little red parts over there that are attached to the lens. Again, working with the ciliary muscles to control how biconvex this lens is. So how much it bulges out or doesn't bulge out as much. Then we move on to the lens. So the lens is this yellow circle oval shape over here, and it is biconvex, meaning it bulges out on both sides. And the lens is suited for its job because remember, the light has to go through the cornea, through the pupil, through the lens to eventually go to the retina. Now, your lens has to be transparent, just like your cornea, to allow light to go through, to enter past. And it's biconvex, which we know allows for refraction of light rays, so it allows the light rays to be bent. And it's elastic because the suspensory ligaments are attached to it. It can change the shape of the lens, allowing more or less refraction to take place. And again, we will discuss that later on how that works. Okay, so once light has gone through, it then goes to the retina, which is the innermost layer of the back. Now, your retina has photoreceptors, and remember, a receptor would detect the change in the environment, so would detect the stimulus and generate an impulse. So the receptors in the eye are called photoreceptors. Photo means light, and because we're dealing with light, it makes sense, and then the receptors. And we have two types of photoreceptors. We have rods and cones. Rods deal with black and white, so seeing colors in black and white and the intensities. So will work typically at night. So if you are walking around the house at night and you don't want to switch any lights on to wake up anyone, then your rods would be very stimulated. And then you've got cones, and your cones is going to be for color. So always think cone C for color C. So they'll be stimulated when we are in the daytime, seeing color. Now, on the retina, we have two spots. We have a yellow spot. So the yellow spot will consist mostly of cones, and when the light falls on the yellow spots of the retina, it will form the clearest, sharpest image. So the yellow spot forms the clearest, sharpest image. And then we also have a blind spot. So just like when you drive and you get a blind spot, same thing here. And the blind spot here has no photoreceptors. So if the light does end up falling on the blind spot, there are no photoreceptors there to generate an impulse, so you won't be able to see anything. Okay, so once the photoreceptors have generated an impulse, then it gets sent along the optic nerve, and that will go to the brain. And because we are dealing with sight and being able to see, what part of the brain will it go to? It would go to the cerebrum. So it's always important when they're asking questions or it needs to go to the brain that you specify what part of the brain it goes to. So because it's sight, it goes to the cerebrum. All right, so that is the basic structure of the eye. So just remember, the light travels in straight lines. It will go through the cornea, which is convex, meaning it will refract the light rays, and it is transparent. It then goes through the aqueous chamber, through the pupil, and the size of the pupil is being controlled by the muscles in the iris. It then goes through the lens. The lens is also transparent to allow the light to go through, but it's also biconvex. And that amount of convexness, the bulging of it, is controlled by the suspensory ligaments and the ciliary muscles in the ciliary body. And that light would then fall on the retina and the photoreceptors would be stimulated to generate an impulse which goes along the optic nerve to the cerebrum. Okay, so two processes that you do need to know when it comes to the eye. The first one is pupillary mechanism. So how does our pupil change size and why does it happen? So if we look at our eye, so we can see in this picture over here, the dark part, the black circle, is the pupil, and surrounding the circle would be the iris. Now, the iris consists of two muscles. You have your radial muscles, and think of a radius of a circle, that's half the diameter. So those are the ones that are going out, the radial muscles. And then you have your circular muscles, and circular, it's going around the eye. So when we are in dim light, that's like a dark environment. Like I said, you're walking around the house at night, you don't want to put the lights on to disturb anyone, or you're at the movies. What will happen is your pupils will dilate, so they get bigger. How does that happen? Well, your radial muscles and your circular muscles work in opposite directions. So they work antagonistically. So to get our pupil dilated, what will happen is your radial muscles contract, and literally think of the radial muscles shortening, and it's pulling the pupil open. That's the way I envision it. And if your radial muscles contract, that means your circular muscles relax because they work oppositely. Your pupil dilates, and the reason why do we want our pupil to dilate? It's so more light can enter the eye. Then, if we are in bright light, we don't want to damage our photoreceptors, so our pupil will get smaller and it constricts. How does that happen? Again, you've got your radial muscles and you have your circular muscles, and they work oppositely. So for this case, the pupil, we want it to become smaller. Your circular muscles of the iris will contract, and therefore your radial muscles relax. Your pupil becomes smaller, it constricts, and the end result would be less light enters the eye. Now, this whole process is a reflex action. So it just happens, it's an involuntary action, it's a quick response to the amount of light in the environment. Right, so that's pupillary mechanism. Very quickly, so this is the wording for it. This is available in the Mind the Gap textbook, which is online. So if we're looking at bright light, so remember the end goal is that the pupil constricts. How does that happen? or your radial muscles of the iris, important you mention that, relax. Your circular muscles of the iris contract, your pupil constricts, and the end result, this is your fourth mark that some students tend to leave out, is that less light enters the eye. So remember, I always tell my students, you got to finish the story off. And then when light is dim, so this is in darker situations, we want our pupil to dilate. How does that happen? Your radial muscles of the iris contract, circular muscles of the iris relax. Remember, they work oppositely. Your pupil dilates. And why do we want that to happen? What is the end result? So more light enters the eye. All right, so just learn that word in as is. Then we're going to go on to accommodation. So accommodation has to do with the shape of our lens and allowing us to see objects far away and then close. So it works between 6 meters would be close and further than 6 meters would be far away. All right, so how is accommodation working? So accommodation, if we look at this situation over here, this is when we are viewing things far away. So our end goal is to get our lens less biconvex. So we don't want it to bulge out as much. If you look at this picture, you can see it's less biconvex. And that means if it's less biconvex, less refraction of light rays will take place, and the end goal is so that light rays can fall on the retina to stimulate the photoreceptors. Right, so that's how that one happens, and I'll show you on the slide in detail how we bring about that change. And then when we're viewing objects nearby, so for example, you read in a book, what will happen is you need to get your lens more biconvex. So in other words, it needs to bulge out more. And if it bulges out more, that means more refraction of light rays will occur, and that means that the light rays will fall on the retina to create a clear image. Right, so how do we bring about these changes? Again, this is from Mind the Gap, so just learn it as is. So if we are viewing objects further than 6 meters, that's far away. Like I said to you, the goal is to get our lens less biconvex. So how does that happen? Well, we have our suspensory ligaments and our ciliary muscles working on it. Now, our ciliary muscles are muscles, so we use the word contract or relax. Our suspensory ligaments are not muscles, so you cannot use the words contract and relax. So instead, we use the word becomes taut, or you can say tightens and slackens. So that's very important that you use the correct terminology here. All right, so how do we let the lens become less biconvex? Well, firstly, your ciliary muscles relax. And then your suspensory ligaments tighten or become taut. So imagine these are strings on your lens and they're becoming taut. So they're pulling on it. And you can see if they pull on it, it would then make the lens become less biconvex. Then the tension on the lens increases. And as a result, the lens is less biconvex. And because it's less biconvex, your light rays are refracted less. And again, finish the story off to get your 6 mark. That means the light rays will be focused on the retina to form a clear image. If we are viewing objects nearby, so that means we want our lens to become more biconvex. How do I let that happen? Well, your muscles are going to contract. And my suspensory ligaments, if you look here, they slacken, so they lessen the tension on the lens, and that automatically causes the lens to bulge out more and to become more biconvex. So your suspensory ligaments slacken, the tension decreases on the lens, and the lens becomes more convex. If it's more convex, the light rays are refracted more, and end the story off, the light rays would be focused on the retina to form a clear image. If this process doesn't happen, then the light rays will not fall on the retina and we won't see a clear image. We would then see a blurry image. All right, so that is accommodation. If we move on to visual defects, like I said, there are 4 that you need to know. So the first one is short-sightedness. So this is when you can see near, but seeing further away, it's blurry. So what happens here is either the lens can't become flat, so can't become less biconvex, or your eyeball is too long. And instead of the focus point here, which should fall on the retina, it's fallen before it. And therefore, it creates a blurry image. So how do you fix this? So you fix this by wearing concave lenses. So you'd wear glasses that have a concave lens. It's important to know how to treat it. So short-sightedness, they can see near, not far. Why? Their lens can't become flat or their eyeball's too long. So the focus point is fallen before the retina, not on the retina. And if you use a concave lens, it fixes it so that the image will fall on the retina. Long-sightedness means you can see far, but you can't see near, so you can't see close up. So this would be when your lens can't become more convex, so can't bulge out more, or your eyeball's too short. So you can see here the light rays are coming together after the retina. It should come together here, but it's coming together afterwards. So that means you're not going to form a clear image. How do you fix that? Well, you fix it by wearing convex lenses. So that will add extra refraction and that will allow it to fall on the retina to produce a clear image. Then the other 2 is astigmatism. So this would be astigmatism, and this is due to the uneven curvature of the cornea or lens. So you almost creating multiple focus points, and then you see it's blurry vision compared to normal vision. And then our cataracts. Cataracts is the 4th one. So cataracts is when it becomes very cloudy, your lens, and that would mean that light can't pass through properly. And again, a blurry image is formed. All right, so that's the eye quickly. This is not in your workbook that you've got. I want to quickly go through this just so you can see how the questions can be asked, and then we're going to move on to questions in your workbook. All right, so this is showing the eye under different conditions. So we've got A, B, C, and D. The first question says, name the process that occurs when the curvature of the lens changes to focus on a near or distant object. So anything to do with the lens changing, that would be accommodation. And the next one says the process in which the pupil size changes to regulate the amount of light entering the eye, that would be pupillary mechanism. Okay, then the next one says, give the letters of 2 diagrams that represent the condition of the eye of a person in dim light. So in dim light, that's darker light, you would want to increase your pupil size. So remember, this is going to be your pupil. This gap here is representing the pupil. So if we want to increase the pupil size, because it is in dim light, we want it to dilate, which 2 letters represent that? So which 2 letters are the pupil the largest? And your answer here would be B and D. Sorry, I made a mistake here. It's going to be B and D over here. Okay, when the pupil's the largest. Then when you're focusing on a distant object. So that means you're looking further than 6 meters. So we need to look at our lens when we're looking at distance. So we're looking at the lens shape. And because we're looking far away, we want our lens to be less biconvex. So if we compare the two, A and B, the lens is less biconvex, meaning it's bulging out less. If you look at this shape compared to this shape, you can see it. Then it says, give the letters of two diagrams that represent the eye of a person whose ciliary muscles are contracted. So the ciliary muscles, that's going to happen in pupillary mechanism. And when they are contracted, sorry, I made another mistake here. When they are contracted, ciliary muscles is got to do with accommodation. So that is going to be sitting up here. It's in your ciliary body. So when they are contracted, that means the suspensory ligaments slacken and the lens would bulge out more. So where would it be bulging out more the lens? That would be C and D. And then where the radial muscles are relaxed, so this would be when the pupil is going to be constricted because your circular muscles are going to be contracted. And where's the pupil constricted? Well, it's the smallest by A and C. All right, so let's start to practice. So in your workbook, please turn to page 14. And you're going to start with question 3.1. I'm going to give you about 10 minutes to complete this, and then we will discuss it together. I'm going to give you a few more minutes and then we will go through it and discuss it. Okay, so let's start to go through this question. So here we've got the diagram below represents the structure of a human eye. So before I start to look at the questions, I start to analyze the diagram that I've gotten. So they've given me a label iris and they've given me other labels. So I start to automatically fill in those other labels just so I can get my head around what we are dealing with. So if we look at A, so A would be a part over here in the retina that would be where the optic nerve leaves to go to the cerebrum. So A would be your blind spot. B is pointing to this structure over here, so it would be your lens. C is the front part of your eye and it's curved, so it's convex, so that would be your cornea. And D would be the back part of your eye, and you can see it's the outermost layer, which is the sclera. So there, just before I even look at questions, I quickly pop down what those labels are. And remember, your back part of the eye consists of 3 layers. You've got your sclera, your choroid, and then your retina. All right, so now we start looking at the questions. So they say identify part A. So we've identified that part A is the blind spots. Part C, we've identified the front part of the eye that's curved, so it's the cornea. And then identify part D, which is the outermost layer of the eye, so that would be your sclera. Then it says, describe, so that's an important word, how the muscles of the iris enable a person to see in dim light. So as you can see, as I'm reading through the question, I'm underlining, or you can highlight key words which will help me answer it. So we're describing how the muscles in the iris enable a person to see in dim light, and it's for 4 marks. So you almost need 4 facts down. So because it's in dim light, the goal for the pupil is to cause it to become bigger, to dilate. So how do we have that happen? So let's look at how we answer it. So remember, we've got 2 muscles in the iris and they work oppositely, antagonistically. So your radial muscles will contract, your circular muscles will relax, and as a result, your pupil dilates. So that's going to get you your 3 marks because we're describing how the muscles in the iris allow a person to see in dim light. So we spoke about the 2 muscles. Radial muscles contract, circular muscles relax. And remember, I picture the radial muscles are pulling the pupil wider. That's just how I remember it. What happens? The pupil dilates, but it's a 4th mark that we still need to get. Why do we want this to happen? How is it allowing the person to actually see in dim light? Well, because the pupil dilates, then more light can enter the eye. So it is very important you always end the story off. So we're describing how the muscles are working and how the person sees in dim light. So the end result, more light enters the eye. All right, then if we look at the next one, name the process that occurs in the eye when a person focuses on objects at different distances. So this has to do with us seeing near and then far. And what is that process called? Remember, we have 2 processes in the eye you have to know, pupillary mechanism and accommodation. This has to do with distances, so it is accommodation. And then 3.1.4, explain how the shape of part B enables a person to read a book. And it's 3 marks. So it's an explain question. It's cause and effect. What happens? Why? That's how I've got to think it. You've got to think of cause and effect with this one. So explain how the shape of part B enables a person to read a book. So they're not asking you to describe accommodation. They want to know how part B, which is the lens, allows us to read a book. Reading a book, you're going to read a book at a short distance. So that means my lens needs to become more biconvex. It needs to bulge out more. So that's how I'm going to start because we're talking about the lens and how it allows me to read a book. So you get a 1 mark for saying it will be more convex. So the lens will be more convex. But how does that allow me to read a book? Well, because it's more convex, that means your light rays are refracted more. And if that is refracted more, it means that it will fall on the retina, the light rays, and focus and form a clear image on the retina. So for this one, like I said, remember they didn't ask you to describe how a person can read a book, because then you would talk about the ciliary muscle and the suspensory ligaments. They wanted to know how the shape of the lens allows a person to read a book. So like I said, you dissect it, reading a book is close by. So how does the lens enable it? It becomes more convex, it bulges out more. And what does that allow? The light rays to be refracted more. And how does that allow me to read a book? Well, it's so that the light rays can be focused on the retina to form a clear image. Okay. So that is how we would break apart that question and how we answer it to get to our 3 marks. Okay, I'm just going to leave this slide on for a few more seconds in case you need to copy down anything, and then we'll move on to the next question. Okay, so you're now going to do the next question, 3.2. It is on page 15. Please complete this one. I'm going to give you 5 minutes to do it. So I'm just going to go back in case anyone needs to copy down. But if you are done copying down, please go on to 3.2, and I'm giving you 5 minutes to complete it. I'm going to give you another minute or so to answer this, and then we'll go through it. Okay, so let's discuss this one. So 3.2, the diagrams below show the response of the human eye to two different conditions. So if we look at diagram 1, what I'm noticing here is that the pupil size is changing. So in this one, the pupil is dilated, it's bigger. And then in this one, your pupil is smaller, it's constricted. So this condition, what environmental condition would it be when your pupils dilated? So that will be in dim light, so darker setting, like I said, a movie house, etc. This one, your pupils constricted, it's smaller. So this would be when it's very bright lights. So that's diagram 1. Then if we look at diagram 2, what I'm noticing here, you can see the pupil size almost remains the same. So we're not looking at the pupil size over here. We're not looking at pupillary mechanism. Instead, what I'm seeing a difference is in the structures over here, the lens and the suspensory ligaments, and then the ciliary body. So I can see here the lens is less biconvex, and over here, the lens is more biconvex. It's bulging out more. And you can also see the difference on the suspensory ligaments. Here it's taut, it's tightened, the tension's increased on the lens, and here the suspensory ligaments have slackened, so the tension's decreased, and that's caused it to bulge out more. So this would be this lens shape being less biconvex would be when we are viewing objects far away, and this one would be when it's more biconvex when we're viewing objects nearby, less than 6 meters. All right, and then you fill in your labels. This is before I even look at the question. So if I had to quickly fill in the labels, you would have the outermost layer, which is the sclera. In the front, it becomes the cornea. You've got the lens, and you've got your ciliary body. All right, so let's look at the question. So 3.2.1 says identify part A. So part A would be your sclera. Remember, that's the white part of your eye, tough, inelastic, basically muscle tissue that will allow the eye to stay in shape. Then you have B, which is the lens, which will allow light to pass through and to refract it. And C is your ciliary body, which contains your ciliary muscle, which will control the shape of the lens. It is important that you know these functions, not just the labels. All right. Then 3.2.2 says identify the process in diagram 1. So because we could see that the pupil size is changing, that would mean that the process is going to be pupillary mechanism because it has to do with the pupil. Then 3.2.3, name, so not a letter, name the part of the eye that is responsible for the response in diagram 1. So what part of the eye is controlling the pupil size and therefore controlling how much light enters the eye? Remember I said sometimes they like to trick you out on this one. So the part of the eye that's controlling the pupil size and controlling the amount of light is the iris. And in the iris, we have the muscles, the circular and the radial muscles that is controlling it. But they want to name the part of the eye, iris. Don't get confused with that one. Don't let it trick you out. Then 3.2.4, state the consequence to a person's vision if the process in diagram 2 does not occur. So how will the person's vision be affected if this process is not happening? Now remember in diagram 2, we have a less biconvex lens, so flatter lens, which is for far objects, so seeing things far away. And it's going to a more biconvex lens, so it is becoming, it's bulging out more. Okay, so that's what's happening over here. But now they're saying state the consequence if this process doesn't happen to the person's vision. Well, then what would happen to the person's vision is that they would see blurry images when they're looking nearby, or only distant objects, far objects, will be clear. So there are two ways you could have answered this. But remember, you need to answer the question, the consequence to the person's vision. So you either say that near vision will be blurred because this process can't happen, or you can say distant objects or far objects will be clearly visible. Only distant objects will be clearly visible. It's important you put that only there, that you emphasize and they can only see distant objects clearly and not near. So it's how this is affecting their vision, the consequence for their vision. Right, so please make sure you actually answer the question correctly. All right, so that's that one. Let's go on to 3.3. I'm going to give you 10 minutes to answer that one. So you got to go to this question, please. I think it's in page 16 in your workbook. If not, just find 3.3. So while you answer this, I'm just going to leave this other PowerPoint open, the slide open, if you need to still copy down, and then I'll change over in a few minutes. So you're now doing 3.3, and I'm giving you about 10 minutes. I'm going to give you a few more minutes. Just remember, read the question carefully and let the mark allocation guide you. So remember, you need 4 facts for them if it's for 4 marks. Okay, let's go through this one. So 3.3 says the diagrams below represent the pupillary mechanism in the human eye. So pupillary mechanisms, they're telling you what you are looking at. So we have diagram A and diagram B. So diagram A, what I notice about the structure X, it's much smaller than the structure X in B. So you can see it's constricted, and this would be dilated. And then we know that is the pupil. Y would then be the iris. And remember, the iris is controlling the amount of light that enters the eye. So just put those labels in for you. So X would be the pupil and Y would be the iris. And if they ask this, like let's say they did that, that would then be the sclera. Okay, so just that's a side note. So 3.3.1 says, identify part X. So we've identified the pupil, Y is the iris. Then the next question says, explain. So it's an explain question why the pupillary mechanism is considered to be a reflex action. And that is for 3 marks. So here you need to think, why is it a reflex action? So remember, a reflex action is a quick response to something. So think of you blinking, sneezing, your pupils dilate. And so it's a very fast response. And it's an involuntary response, meaning you don't have conscious control over it. And pupillary mechanism, what are we responding to? What would be the stimulus? Because your pupil size is changing to control the amount of light entering. So the stimulus here would be light. Okay, so how do we formulate that, what I've just said, into the correct answer? So I'm just pulling it up over here. So remember, it's an explain and why it's a reflex action. So why is pupillary mechanism a reflex action? So you would start off by saying it's a rapid. They would also accept quick, and it's an involuntary response. So you don't remember have conscious control over it. So it's a rapid or quick involuntary response. And why is pupillary mechanism it? So your 3rd mark is going to be because it's to a light stimulus. If you just responded with the first 2 points, you basically just saying what a reflex action is, which is a rapid involuntary response. And we know it's there to protect the body from harm. But you need to link it to why pupillary mechanism is a reflex action. And that's because it's a rapid involuntary response to a light stimulus. And that will get you your 3rd mark. So cause and effects for this one. Right. 3.3.3 says name the 2 effector muscles that are involved in the pupillary mechanism. So these are muscles involved in pupillary mechanism. So that'll be the muscles of the iris. But what are those 2 muscles called? 2 marks. It's going to be your radial muscles and your circular muscles. Please do not get confused between circular muscles and ciliary muscles. Remember, ciliary muscles has to do with the accommodation, so controlling the shape of the lens. Circular muscles is in the iris, and so are radial muscles. So the radial are the ones that go out, and the circular are the ones that go around. Sorry, it's hard to draw that. Okay, so that's the answer for 3.3.3. 3.3.4 says, again, it's an explain the significance of a change in diameter of part X from diagram A to diagram B. So the significance is the importance. So why is it important for the pupil diameter to go from a small one to a big one? That's essentially what you need to describe, not how it happens. So not describe the change, in diameter. That's not what they're asking. They're asking, explain the significance of the change. In other words, why it is important it undergoes that change. All right, and it's for 4 marks. So this is where you need 4 facts. So let's look at the memo answer. So firstly, talk about the change. What is the change happening with structure X? So we can see it's was constricted and it's becoming dilated. So that will be your first mark. But first telling me the change, and then we're going to go on to explain why that's important. So 1 mark for saying the pupil dilated. Why does the pupil dilate? What is the purpose of the pupil dilating? And that is so more light can enter the eye. So I'd hope that you at least got those 2 marks already. But now this is where you need to take it a step further and just explain it a bit more in detail, the importance of that. So the pupil dilated so more light can enter the eye. Why do we want more light to enter the eye? Think when it's dark. Why do we want more light to enter the eye? Obviously, so we can see better. So that would be your third point. So more light can enter the eye to improve vision. Also, we can see better. And in what condition, environmental condition, does this happen in when our pupils dilate? In dim light. And that's what gets you your 4th mark. So if we just look at the question again, it's saying explain the significance in the change in the diameter of part X from A to B. So yes, the pupil dilates. Why is the pupil dilating? So more light can enter the eye. But why do we want more light to enter the eye? Why is that important? So we can see better, so it can improve our vision in dim light. So we're ending off in the condition. So that's going to get you your 4 marks for that one. So that's why I said it's very important you read in that question carefully. If you describe pupillary mechanism, you most likely will only get 2 marks. You didn't then describe why it is important that it does dilate. Okay. So let's go on now to question 3.4. I'm going to give you 5 minutes to do it. So this is a question you're doing. I think it's still on page 16, 3.4. I'm just going back here in case anyone needs to still copy down the answers. So 5 minutes to do the next question. I'll go to that slide now. I'm going to give you another minute or so, and then we'll go through this one. Okay, so this question has to do with our visual defects. That's why it's so important that you understand the visual defects properly. So 3.4 says a man is long-sighted. So remember, long-sighted, they can see far, but they can't see near. So the first question, 3.4.1 says, explain how the structure of his eyeball affects his vision when he reads a book without glasses. Okay, so there's quite a bit of information in there, and it is for 3 marks. So firstly, it's an explain question. Secondly, it's about the structure of the eyeball. So you cannot talk about the lens because it didn't ask about the lens. They want the structure of the eyeball and how it affects his vision when he's reading a book. And when you read a book, you are near. Now remember, he is long-sighted, meaning he can clearly see far, but not close. Close would be blurry. So how does that eyeball result in blurry vision close by? So let's look at it. So I've used this diagram here because it's nice to show it. So this guy has the bottom eyeball. So if we look, the eyeball is shorter than this one. So this is a normal eyeball. This one is shorter. So what is happening in a normal person who can see close up? The light rays are going to come together and fall on the retina. Okay, what we want, and that produces a clear image. However, because this man's eyeball is shorter, the light rays will come together outside at the back of the retina, so behind the retina. And that means when it hits the retina over here, it hasn't come together and it's not going to form a clear image. So that's why they produce or see blurry images when they are viewing things closely. All right, so how do we explain it? So I just want to underline the key words. So just explain how the structure of the eyeball affects the vision when he reads a book. All right, so the structure of the eyeball. So firstly, you're going to say that the eyeball is too short. Remember I said you cannot talk about the lens because that was not the question. So you start off by saying the eyeball is too short. Therefore, what happens? Well, the image is going to form behind the retina. So instead of the image forming on the retina to produce a clear image, the image is going to form behind the retina. And how do you end it off? How does it affect his vision, this? Well, it will cause blurred vision when he reads a book. All right, so you had to almost break it down. Firstly, you're talking about the eyeball. In this case, it's too short. Therefore, what happens? Well, the image forms behind the retina. And how does that affect his vision? Well, it causes blurred vision. Okay, so that's how you would answer 3.4.1. Then we go on to 3.4.2. So this says, Explain how wearing glasses with convex lenses will improve his vision when he reads a book. And that's for 2 marks. So how does convex lenses improve his vision? Remember, convex means it bulges outwards. Sorry. Concave will mean that it goes inwards like a cave. But we're looking at convex, so we're looking at how it bulges outwards. So how does that lens improve his vision when he's reading a book? And remember, when you read a book, that is close up. So that's near vision. All right, so let's have a look at how we would answer that question. So with this one, so here you can see in the bottom picture, he is wearing a convex lens, and I say to you now, convex lens bulges outwards. If it is convex, bulging out more, what does that mean for the light rays? Well, that means the light rays would be refracted more. So an extra layer of something being convex or bulging out more will refract the light rays more. So that's your first point. The light rays will be refracted more. And how does it improve his vision? Well, if it's refracted more, that means that the image will fall on the retina. So if we look at the top image, here, without the lenses, the image falls, remember, behind the retina. But having the correct treatment, in this case convex lenses, it causes extra refraction of the light rays, and that will allow it to fall on the retina, and the image will form on the retina. So that is how you would answer that one, because they want to know how the convex lenses improves his vision. All right, so those are all the questions we are doing on the eye. We're going to move on to the ear now. So before we do that, just a reminder, with the eye, it is so important you know how to label it, you know the functions, you know pupillary mechanism, accommodation, and of course, you know your visual defects. And that's the 4 that we've done. And you can see this was a 5-mark question on visual defects. Okay, so let's go on to the ear. So I'm going to first do a quick recap, and then we'll start our questions. So the ear, if we look at the ear, it is important that you know also the structures and the functions. So we divide it up into 3 sections: outer ear, middle ear, and then the inner ear. So if we start off, we have the pinna, and that's what you physically see on the outside. So that's the little flap. And what that does is it directs the sound waves to the auditory canal. And the auditory canal basically is just transferring those sound waves to the middle ear. We then have a little membrane called the tympanic membrane. And that also you may know as the eardrum, but we call it the tympanic membrane. So try stick to that word, please. So once the sound waves hit the tympanic membrane, it causes the tympanic membrane to vibrate. So almost imagine it like a piece of paper. And if you flick that piece of paper, you're going to cause the tympanic membrane to vibrate. And that thing passes along the vibrations in the middle ear. So here it will go from ossicle to ossicle to ossicle. So there are 3 ossicles in the middle ear, arranged biggest to smallest. So we have the hammer, the anvil, and the stirrup. But you can use malleus, incus, stapes, whatever, whichever one you want to use, that's will be fine. So the vibrations are then passed along the ossicles, which would then amplify it until it gets to the oval window. And that then will cause our oval window to vibrate, and that creates pressure waves in the cochlea. So we had sound waves in the pinna and auditory canal. The tympanic membrane creates vibrations, which goes along the ossicles to the oval window, which would then create pressure waves in the cochlea. Those pressure waves will stimulate the organ of Corti with the hair cells, which are the receptors, to generate an impulse, which would then leave the cochlea and then go to the brain. And because we're dealing with hearing, what part of the brain would the auditory canal take the impulse to? For hearing, it will go to the cerebrum. Then we also have the round window. So the round window is going to absorb any pressure waves, so it doesn't bounce back and distort our sound. So instead of that happening, it absorbs the pressure waves and doesn't allow an echo to happen. And then we have the eustachian tube, which basically connects our ear to the back of our throat, and that's there to equalize the pressure in our ear. Okay, so that's the basic structure of the ear. What I want to just bring to your attention and emphasize again, it is so important you use the correct terminology when you are describing hearing. So it's sound waves, then vibrations, then pressure waves, which would then generate an impulse, which would then get sent to the cerebrum. So how you would describe that? So the pinna would conduct the sound waves to the auditory canal, and that would cause the tympanic membrane to vibrate. Vibrations are passed along the ossicles and they are amplified and they're taken to the oval window, which will vibrate and create pressure waves in the cochlea. That will stimulate the hair cells on the organ of Corti to generate an impulse, which will then be taken along the auditory nerve to the cerebrum. So please take note. First sound waves, then it becomes vibrations in the middle ear, and in the inner ear, you have pressure waves. So that's very important that you know that. Then if we look at the vestibular apparatus, so the cochlea is involved in hearing. And like I said, the impulse generated there will go to the cerebrum, part of the brain. Your ear is also involved in balance, and that's when your vestibular apparatus comes into play. So that consists of your semicircular canals, which are these three circle-like structures, which end off in ampulla. And in your ampulla, you're going to have receptors called cristae. And I'm going to discuss that now. And then you have two bumps almost, your utriculus and sacculus, which will have other receptors called maculae. Together, this whole structure is going to help with balance. And that will also go along the auditory nerve to what part of the brain? What part of the brain is involved in balance? Well, that's going to be the cerebellum. Okay, so let's quickly look at how we maintain balance. I know I'm going through this a bit quickly, but you should have been taught this already. So I'm just doing a quick recap so we can get into the questions. All right, so how do we maintain balance? So if they ask for change in direction and speed of the head, so we change in direction and speed of the head. So almost you're looking around, you're playing a song, imagine you're looking from, you're running, you're looking from side to side to side. How are you making sure you don't fall over and that you maintain your balance? So when there's a change in direction and speed of the head, the cristae is found in the ampulla in the semicircular canals. So the cristae would be stimulated because it's a receptor, generate an impulse, which will then send it along the auditory nerve to the cerebellum. Right, so the cristae is found in ampulla, which is in the semicircular canals. How do I remember the receptor? Well, semicircular canals has those C's for cristae. And how do I remember that whole thing? by the little acronym acids. So ampulla contains the receptor called the cristae, which when stimulated will generate an impulse and then will send it to the cerebellum in response to changes in direction and speed of the head. Okay, so that's the one part. The other part is it's responding to a change in position of the head with regards to gravity. So for example, if you're upside down, this would be then when these receptors would be stimulated. So the receptor for this one is called the macula, and that is found in the sacculus and the utriculus. So when your head changes position, the macula and the sacculus and utriculus will be stimulated to generate an impulse that would be sent along the auditory nerve to the cerebellum, which would then help with the balance. How do we remember this one? So we remember it by SUMP. So the S is for the sacculus, the U for utriculus, the M for the receptor, which is macula, and then it's for your position of head with regards to gravity. It is so important that you learn this. They like to ask this question, how do you maintain balance? So please learn the little acronyms and then just learn those full sentences because it can be worth 4, 5, even 6 marks sometimes. So we don't stop there. Then you need to take it a step further, and it's the same for both situations. So the receptors receive the stimulus, convert it to impulse, send it along the auditory nerve, and it's to the cerebellum. Why? Because we're dealing with balance. Hearing goes to the cerebrum, balance, cerebellum. And then we're not done yet. This would then send an impulse to the muscles to maintain balance. So it's going to the effectors to maintain balance. Okay, so it is important that you learn these. Remember, this is applicable for both situations. All right, and then just ending off this section before we practice with just hearing defects. So there are 2 hearing defects that you must know, and how do we essentially treat it? So we have hearing defects. So the first one is middle ear infection. So here you might have excessive fluid there, or your pressure's not being equalized. So they insert a little tube and it's open called a grommet. So the grommet is inserted into your tympanic membrane, and that then will allow fluid to drain out your middle ear, and it also helps to equalize any pressure. Deafness. So for deafness, this could be where parts of your ear has been injured, specifically your inner ear, your cochlea, or it could be when you've got hardened wax or your ossicles are hardened, so they can't vibrate. And if they can't vibrate, then your oval window can't vibrate. And if your oval window can't vibrate, then pressure waves can't be created. And if pressure waves can't be created, then your organ of Corti won't be stimulated, no impulse generated, and then you can't hear. So how do we fix that? So we use a hearing aid to fix that. So if I just show you on this picture, it's easier. If your ossicles over here are hardened, so they can't move, they can't vibrate, just think, well, if that can't pass the vibrations along, that means your oval window can't vibrate, which means pressure waves can't be created in the cochlea, which means the organ of Corti won't be stimulated. And then, like I said, impulse won't be generated, and then you can't hear. So you got to almost take it with the ear, step by step by step. I often find students, like I said, don't finish the story. You miss out on steps. You need to go, this doesn't happen, that won't happen, that won't happen, etc. Okay, so let's start practicing. So if you can turn, I think it's on page 17, to question 3.5. I'm going to give you about 8 minutes, 7 to 8 minutes to complete this one. I'll give you 2 more minutes and then we'll go through this one. Okay, let's go through this one. So here we have a diagram which represents a part of the human ear. So this is combining the two pictures I showed you prior. So we've got, this is mainly our inner ear part. So let's start off by trying to label it. Right, A, we've got our three semicircles. So these would be your semicircular canals. B, you can see is leaf and the cochlea, which is C. So that would be the auditory nerve. C, I've just said, is the cochlea. Then we have D and E. Now, remember, shapes do help you, but they don't always draw it to scale. So the first one is going to be the oval window, which is E, and D is going to be the round window. So if we just look at this, we have, I always remember O comes before R. So besides relying on the shape, you also have that way of remembering it. So the oval window is E, and D is a round window. And then F is the little bumps at the bottom, which will be your ampulla. And sitting in your ampulla would be your receptors that belong to the semicircular canals, the C ones, which would be the cristae. All right, let's look at the question. So 3.5.1 says identify parts A and B. So I've done that now for you. We have A, which is our semicircular canals, and B would be your auditory nerve. Then 3.5.2 says give the letter and name. So please don't only write down one of them. Please make sure you write on the letter and the name of the part that creates pressure waves in the fluid of the inner ear. So remember, the vibrations go to the oval window, which would then create the pressure waves in the cochlea, and then any excess pressure waves would be absorbed by the round window. So your answer for this one, creating the pressure waves, 2 marks, letter and name, 1 mark for E and 1 mark for the label, which is oval window. Then they want the letter and name of the part that absorbs excess pressure waves in the inner ear to prevent the formation of an echo, 2 marks. And remember, letter and name. And this one would be D, 1 mark, round window, 2nd mark. Okay, so just remember those two functions. And then 3.5.3 says name the parts of the brain that interprets impulses from part F. Now, F, ampulla, and remember your cristae is sitting in there. And this is all involved in balance. So what part of the brain interprets balance? Well, that's going to be the cerebellum. If they asked you what part of the brain interprets impulses from C, the cochlear part would be for hearing, so that answer would be the cerebrum. But they asked you from F, so that is going to be balance, and they'll go cerebellum. And then they said, name the receptors found at C. So C is a cochlea. What are the receptors in there? Well, it would be the hair cells of the organ of Corti. So the organ of Corti consists of the hair cells, and it's important to know it's the hair cells that are going to detect the stimulus and generate an impulse. And like I said, because it's in the cochlea, because it has to do with hearing, that will go along the auditory nerve to the cerebrum. So there are 3 receptors in the ear. We have the hair cells, which are on the organ of Corti, and that is in the cochlea, involved in hearing. You have your cristae, which has to do with your speed and direction of head, and that's sitting in your ampulla. And then you have your macula, which is sitting in the utriculus and sacculus, involved with position of head. and they are all involved in balance. Okay. Right, let's go to the next question and the last question for the day, 3.6. So I'm going to give you about, I'm going to give you about 7 minutes to complete this, just so that we've got time to go through it at the end, and then just to end things off for the session. So 3.6, if you can do that for me quickly. Okay, I know you probably are not done with this, but I do want to just go through the answers with you so you have it for when you are preparing for your exam. So let's look at this question. So it says the diagram below represents part of the ear. So I apologize, my slide is not showing the first few questions, but it's over here. So the first one says 3.6.1, identify part A. So part A, you can see, is leaving the inner ear, the cochlea, and it's going somewhere. We know it's going to the brain. So A would be the auditory nerve. Please do not get confused between auditory and optic nerve. Remember, the auditory nerve is in the ear, auditory is hearing sound, and the optic nerve is in the eye. Okay. Then identify part B. So part B is a circle spiral thing, and that would be called the cochlea. Then 3.6.2 says state the function of part C. So you need to know that part C is going to be the round rim window. Remember, you can sometimes tell by the shape. They've been nice in this one. You can see this is round and this one's oval. But remember, I taught you O before R. So the function of the round window is to absorb excess pressure waves. So if they said explain the function, then you would say absorbs excess pressure waves to prevent an echo. But they just want to know the function of it. Absorbs excess pressure waves from the inner ear. And then they said state the function of part D. So part D would be the eustachian tube, and its job is to equalize pressure on both sides of the tympanic membrane. So sometimes when your ears are blocked, the pressure is building up in your middle ear. So you normally try to yawn, so to equalize that pressure, because your eustachian tube, like I said, is connected to the back of your throat. So the function is to equalize pressure on both sides of the tympanic membrane. Because if pressure builds up in the middle ear, what can end up happening if it doesn't equalize it, if the eustachian tube can't equalize it, it can burst the eardrum. Okay, so important to know the labels, but also the functions of the ear. Then we look at the next question, which it says, just want to get it for you. Otosclerosis is a medical condition that prevents the ossicles from vibrating. Explain, so it's explain question, how this condition will affect hearing, and for 4 marks. So how does the ossicles not vibrating affect hearing? So you have to make sure you are answering that question. Will your hearing be improved? Will it worsen? Will they not be able to hear? And why? Cause and effect. That is how you have to answer it. So let's look at the memo answer. So firstly, the person will suffer from hearing loss. That is telling me how it's affecting hearing. Okay, so that's giving you a 1 mark. And the little star there means it's a compulsory mark. So even if you get all the other facts, but you don't have that, then you won't get full marks because you're not answering the question properly of how it will affect hearing. So the person will suffer from hearing loss. Now you need to explain why they're going to suffer from hearing loss. So if the ossicles can't vibrate, then no or less vibrations will be transmitted to the oval window. And if the oval window can't vibrate, then no or less pressure waves will form in the cochlea. And again, it is important that you are using the correct terminology here. First, no vibrations will be transmitted to the oval window, and then no pressure waves will form in the cochlea. And if there's no pressure waves in the cochlea, the organ of Corti won't be stimulated. So the hair cells on the organ of Corti won't be stimulated. And therefore, if it's not stimulated, no impulse will be created and no impulse can go to the cerebrum. And again, you need to mention the part of the brain that it is going to. So here it's important that you explain it step by step, like I was showing you earlier on. If ossicles can't work or can't vibrate, then you go, well, that won't happen, etc. So the vibrations won't go to the oval window. Therefore, no pressure waves will form in the cochlea. Therefore, the hair cells on the organ of Corti won't be stimulated, no impulse generated, and no impulse will go along the cerebrum or go along the auditory nerve to the cerebrum. And then they can't hear because no impulse is taken there. All right. And then 3.6.4, describe. So it's a describe question, the role of the ear in maintaining balance. So essentially, you are now describing the process of it, and it's for 6 marks. They didn't specify if it is speed and direction or position of head. It's balance as a whole. So if I quickly go back, I'll come back to this now. You're describing this entire slide for the 6 marks now. All right. So how do we start? Well, you've got your 2 receptors. So your cristae, which we know is found in our semicircular canals in our ampulla, will be stimulated by a change in speed and direction of the head. So that's the first part. That's the one part of the receptors we're talking about. Let's go to the second part. The macula, which is found in your utriculus and sacculus, will be stimulated by a change in the position of the head. So we've got our acids here, we've got our sump here. Remember the way to remember it. So that's going to get you 4 marks by talking about the receptor and what stimulates them. Then, because they are receptors, they create an impulse. And then we just need to describe how we maintain balance. Well, that impulse gets transmitted by the auditory nerve, and because it's balanced, it is going to the cerebellum for interpretation. And that then will send the impulse to the muscles to restore balance. So when they give you balance, they're not split in the 2, so you have to talk about both of the receptors. And like I said, what stimulates them? So the cristae are stimulated by change in speed and direction of the head. And the maculae are stimulated by the change in position of the head. Creates an impulse, goes along the auditory nerve to the cerebellum. So that's extremely important that you are able to describe that. Okay, so that brings us to the end of today's session. We're quickly going to do a post test. It's going to take you a minute to do it. It's the same one we did yesterday. It's multiple choice. So if you can just scan this QR code, it is in your booklets as well. So if you can just quickly do that, it should take you to this. And if you can quickly fill in those multiple choice questions. So I'm going to give you a few more seconds to quickly do that multiple choice question, and then we'll finish the session. Okay, I just want to say thank you so much for joining today's session and yesterday's session. I hope you learned something new and you gained some valuable tips that you can use in your upcoming examinations. Just remember, the best way to ace life sciences is obviously to study, but then to practice, practice, practice. So to work through past papers and to see how the questions are being asked and how you must answer. I wish you the very best for your upcoming examinations, and thank you once again for joining.